students, I hope all of you are very well, and I am thankful to you all for listening my previous lectures, and I hope with passing of time you are learning a lot and you are entering at the deep of understanding in our immunology, immunity and how our immune system is working. Today I am going to tell you something very interesting which is associated with our immunity, and you can also design new products for our immune system. Today we shall learn about various blood derivatives, plasma, serum, what are the differences between them. Then I will also tell you how to separate serum and plasma, then serum preparation, then some application of this serum, and after that I will tell you in blood grouping how it is important, and overview of various albumin based drugs and albumin as a drug carrier, then some diseases, and with this we shall move for the next lecture. So, these are the very important things we are going to learn. So, let me first tell you blood derivatives. I hope many of you might have seen in your life various kinds of like fresh frozen plasma or plasma protein fractions. So, the question is that how these products are actually being produced and how you will make such kind of products and where you may use. To tell you that I will have to tell you the basics. Here you can see very interesting picture. This is your blood vessel, artery, vein, whatever. If you see in detail, you will be able to see the red blood cell, simply we call it as a RBC. Then you will be finding some platelets, white blood cell and plasma. Okay. If I tell you more with a more zoom and in the classification, I can tell you that this is your whole blood, you have plasma and you have some formed element, leukocytes, platelets, erythrocytes. In previous lecture we have covered this area. Okay. Let me tell you about this part. I hope you can recall in my second lecture on immunity, I have told you in detail about all the various kinds of cell, what are the activity of those cells like neutrophil, basophil, eosinophil and monocytes. You remember I have been taking this part from my that slide. So, you are aware what they are doing, they are actually boosting our immunity, but the fact of the matter is that we also have to consider this part, plasma. We have covered this part, we also have to learn about this part, plasma and including the serum. So, if I focus on this area, I have to tell you that this yellow part, you can see this is actually call it as a plasma. Now, you might be thinking, sir, then what is serum? Let me tell you. So, if you take the entire blood and if you take the plasma and if you remove the clotting factor, then it will be called as a serum. So, serum is the plasma minus clotting factor. It means you have a blood, for example, like this, you have plasma and you have other kinds of substance. If you take just only that entire plasma and if you just remove the clotting factor, then we will be getting the serum. Serum is very important. Here I have a very interesting chart where I can tell you that all the various component whatever we are actually discussing in our previous lectures, whether they are present in whole blood, plasma, serum or plasma water, because ultimately with this all these components are actually coming under our whole blood, right. So, in whole blood you will be getting everything, you will be getting everything, white blood cell, red blood cell, platelets or various kinds of protein, etcetera, lipid, electrocytes. But in plasma you will be not getting these things, in plasma you will be not getting these things, and in serum this clotting factor actually removed. Okay. So, our main area of focus in serum actually relying on these things. Okay. So, let me tell you about these things. Serum. So, it is a clear portion of any animal or plant fluid that remains after the solid elements have been separated out. You can see when you will separate when you are separating, these are the our clotting factor and the remaining serum is actually present here. So, the term usually refers to the blood serum, the clear straw colored liquid portion of the plasma that does not contain fibrinogen or that is blood clots. Fibrinogen, blood clots, various kinds of clotting factor, these are actually responsible for the clotting of blood. If any kinds of wound or any kinds of cut may happen in your blood vessels. So, how it will happen at that time, your all the clotting factor will come to the picture, they will make the clotting. So, for serum or to make some product related to serum, we are actually not considering those clotting factor, we are removing those things. The blood serum from person or animal whose bodies have built up antibodies are actually called anti-serum or human serum, very important, because in the time of COVID this term was so much popular, because the scientists they have tried their best to make some that is a therapy based on this way, that is anti-serum or human serum. So, you can see the inoculation with the such anti-serum provides temporary and passive and immunity against diseases, like example COVID-19, whatever I am trying to tell you. And it is also used when person is already being exposed or contact with a disease. Okay. Then if I tell you more, the diseases in which passive immunization is sometimes used, like diphtheria, tetanus, botulism and gas gangrene. Or in those diseases we are already using this serum. And if I tell that how to separate, how you will separate this serum and plasma, because it is not a very easy task, because you see if somebody got infected with some disease and if antibodies are actually being produced in that person's blood. So, at that time you can use. So, how you will separate the serum and plasma. So, serum is the liquid fraction of the whole blood that collected after the blood is allowed to clot, and the clot is removed by centrifugation, and resulting supernatant designated serum is clearly removed using the Pasteur pipette. So, I hope in your school days you might have done some experiment, you have taken some sample, you have done centrifugation okay, at a certain RPM, when you will do centrifuge all those things. If I can have some picture, if you do centrifuge, you will be getting ultimately this kinds of thing. So, the serum and your blood clots, everything will be get separated. So, after that after that by using the Pasteur pipette you will be just removing your this serum. So, the plasma is produced when whole blood is actually collected in the tubes that are treated with the anticoagulant, and blood does not clot in plasma tube. The cells are removed by the centrifugation, because we already are removing our clotting factors. So, there are no chances of the clotting of the blood in that tube. The supernatant designated plasma is clearly removed from the plate, whatever I mentioned. So, now the thing of understanding is that serum preparation you have that is separated the plasma and serum. Now, how you will that is prepare? So, you have to collect in your test tube. If commercially available tubes are used, then researchers should use the red top tube, whatever I was showing you, and this available in like various companies like Bigton, and they are actually providing. So, you can use their tube. So, after collection the whole blood, it may allow the blood to clot by leaving in undistributed room temperature. So, when it will clotting will be happen. So, obviously, after that when you will do some centrifugation. So, here I have mentioned that usually 15 to 30 minutes you may do for the clotting, and you may remove the clot by centrifugation, that is 1000 to 2000 x gram for 10 minutes. When you will do under refrigerated centrifuge in that certain parameters, then what will happen? It will be completely separated in that your tube, at the top, because serum are actually slightly lighter compared to our the clotting factor. Clotting factor will be that is settle at the value, and at the top you will be getting your serum. Okay. So, the resulting supernatant is designated as the serum. Following centrifugation, it is important to immediately transfer the liquid component into a clean that is a polypropylene tube using a Pasteur pipette, whatever I was actually mentioning you. Then you also have to maintain the temperature, because all these things are actually protein. All of you are aware that at high temperature proteins actually get denaturated. So, we have to maintain the temperature like 22 to 8 degree centigrade while handling. If the serum is not analyzed immediately, then the serum should be that is approbation for 0.5 ml aliquots stored and transported into the 20 degree centigrade or lower. You may try to maintain at as low as possible, so that your serum there should not be at high temperature, they should not be get denatured or some breakage of bonds among the proteins, and they may remain in a very appropriate manner. So, it is important to avoid the freeze throw cycles, because this is detrimental of many of the serum components. Sample which are homolyzed or eccritic or can be certain test. So, you have to understand that some of the diseases, if somebody is suffering from some diseases, so there are chances that some certain components, some that is elements might be present in that sample in serum. So, you have to do some test according to those diseases. Now, where we are actually using, or on those cases we can use these kinds of serum. So, one of the disease ALS. So, you can use this serum from ALS. So, here the anti-serum we actually derived from the animals, and that have immunized against the human lymphocytes. It is a powerful immunosuppressive agents that cause destruction of the circulating lymphocytes. So, in ALS we can use our this technique. If I tell you more, the anti-rabies, you know rabies, if a dog bite a person and if the dog is already infected or exposed with rabies virus. So, there are chances the virus might have that is entered in the person's blood. So, in that case also our these techniques will be helpful. So, anti-rabies serum we can use. So, this anti-serum you can obtain from the blood serum or plasma of animal immunized with the rabies vaccine. If somebody is already vaccinated with rabies vaccine, so obviously in his blood or plasma or serum we will be able to get those antibodies. So, it will be used for the post exposed prophylaxis against rabies, if rabies immunoglobulins are actually unavailable. You can also use in pooled serum. So, this mixed serum as a number of the individuals is another way you can use. Now, another interesting area is actually our blood grouping. So, blood grouping preparations contain particular antibodies that is against the red blood antigens used for the blood typing. So, all of you are aware that we have various kinds of blood types like O negative, O positive, B negative, B positive, A negative, A positive, and AB negative, AB positive. But the problem is that everybody cannot donate blood to everyone, and everyone cannot accept blood from every types of people because of this blood grouping. Here this is the people of those who can donate, and here the people who can recipients. If your blood is actually B positive, then you can that is according to this chart you can take the blood from these people. So, here is a interesting chart, you may take some screenshot and you may save it for your further references, because based on that you can that is understand that to whom you can give your blood. Now, the question is that why it is happening, why the blood group is actually happening. The answer is actually relying on this slide. So, for example, this is the blood type AB, A, B, AB or O. Here is the answer. So, antibodies what are present. So, I have told you in that serum we have we will be getting the antibodies. So, the antibody B, this is present in the group A, and anti-A it is present in the, that is antibody A, it is present in the group B. Here no antibody is present, and here both the antibodies are actually present, that is why it is O. So, based on that, because of the presence of this kinds of antibody, our antigen, this opposite you can see, this opposite. Based on this category, we have different kinds of our blood group. So, that is why our this concepts is highly useful in our understanding. Here I have another very interesting that is slide. Here you can see various drugs. All the drugs are actually albumin based drugs. I will not read all the various kinds of drugs because it will take much time. I have just added just for your understanding that by using these concepts we are actually developing various drugs. I will just mention you that where we are actually using. For example, you can see type 2 diabetes or various kinds of emitting radionuclide imaging agents, or solid tumor we can also use, hepatitis C and other kinds of diseases we can also use our similar technology and all drugs actually available. Now, albumin, that is serum albumin, you can also use as a drug carrier. How? For example, this were albumin. So, by polymerization you can do dimerization, you can make some nanoparticle. You can do protein fusion, and there is some fusion. So, your protein, albumin, both the things will mix and there will be some fused. You can also use these things as a carrier. If you want to inject some protein to some person's body, you can make fuse and you can use this technique for the drug delivery. In case of the surface modification like cationization or glycolization, you can also use albumin. Okay. So, as a drug carrier it can be also used, and serum sickness as an hypersensitivity is very important. So, in many of the cases we have seen that some people having some hypersensitivity. I hope you remember my previous lecture where I have told you about, sorry just a second, yeah, there was some wrong that is a click, that is why I was interrupted. Okay. So, hypersensitivity, I hope you remember in my previous lecture I have mentioned you that hypersensitivity. So, many of the people are actually when they exposed with a certain pyrogen or any kinds of antigen. So, their antibody is actually reacting. Now, again when this you encounter what the person encounter the same substance, foreign substance, his body actually react because of the activity of the immune system. So, if some person actually facing such kind of problem, so we call it as a hypersensitivity, allergic we sometimes we also call. In my previous lecture I hope you remember that concept. Now, another thing I will have to tell you for today, that serum sickness. Okay. So, serum sickness is a hypersensitivity reaction following the administration of the foreign serum or other kinds of antigen. It is marked by unclinical rashes, edema, adenitis, joint pains, high fever and also prostration. Reactions to tetanus antitoxin derived from the horse serum were especially common, but are now rare owing to the refinement of the antigenic component. So, it is a problem, but we can come out from such kind of problem. So, if somebody having some issues with the administration of the foreign serum or antigen, and if they are actually encountered with such kind of various side effects or various reaction, as of now on 21st century we can modify the antigens its structure in our computer. You see in my previous lectures I have mentioned that in silico vaccine design, let me write in silico vaccine design. I hope you remember during vaccine discovery you have mentioned. So, if a antigen or if a serum is actually creating some problem, so we have to change some structure, some structural conformation we have to change or configuration we have to change. We have to change in such a manner, so that it should not make unnecessary binding. You see in our body we have near about that is 25,000 genes, and out of this 25,000 genes they are producing near about 20,000 of the protein, okay, 5,000 are non coding genes. So, those proteins are actually available in whole our body. Now, if you administer any antigen, first all the B lymphocytes, T lymphocytes and etcetera they will be active, and they will try to that is attack on this antigen, including in our body we have lots of proteins also, that 20,000. So, there are chances that foreign substance it might be going to fight or mix with the other our available proteins also. So, that is why in 21st century we are actually using in silico vaccine design. We are actually trying to see off target binding. We try to identify see that our protein of interest, our antigen, it should not go and bind with any off target. So, this is a this problem can be that is solved. Another thing, serum sickness syndrome. So, serum sickness syndrome is a serum sickness like hypersensitivity reaction occurring after administration of certain drugs. it is marked by clinically by low grade fever, then arrhythmia, facial edema, pain, also swelling in the joints, and lymphadenopathy also occasionally may be associated with the nitritis, with the bacterial pulses, and also Guillain-Barré syndrome. So, these kinds of problem you can also face. So, whatever I already have mentioned with you, that yes, we can solve this problem. How we can solve? We can solve by using our same in silico vaccine design. But you have to understand, this technology is actually emerging. If you simply search on Google Scholar, PubMed, or any of these sites, you will be observing, if you see the graph, that number of articles are actually published in last 50 years on certain, on particular these concepts, that is anti-serum concept. You will be observing in last 10 years, or I can say 15 years, there are lots of research papers. So, how the new research papers are coming? Because of the advancement of in silico design, vaccine design you can say, or in silico design. Because of the advancement of computer, you see another thing I like to mention you, that HPC. I think in my previous lecture I also tried to highlight HPC, high performance computing, because when I say in silico, we are actually doing entire work in our computer. We have proteins 3D structure, and we have to, we have lots of docking software. We can do docking of our antigen into with the available proteins for the identify the off-target binding. So, for that calculation, if normal computer you may use, normal PC you may use, it will take long time. Okay. For example, docking, if you do simple one docking, your some B lymphocytes and other things, in normal computer if it is taking 30 days, HPC will can complete this work within 3 days, or it is possible much more earlier time, much more less time, sorry. So, that is why at 21st century we have solution, and if you may think about this, if you may more study about these concepts, you can also solve this problem. So, that is why as a student of B Pharm, I will suggest that you may think about this concept, and so that we can get much more better results. Another thing I like to mention you before ending my this lecture, that you see drug is not a complete solution, because there are lots of diseases like cancer. Nowadays in cancer, the most useful that is therapy is monoclonal antibodies. Monoclonal antibody size is much more bigger compared to our normal that is small molecules based drugs. Okay. So, the problem is that their molecular size is much more higher. So, that is why you cannot take those monoclonal antibodies orally, or either you may have to do some modification, or because there is a issue with your absorption in the intestine because of their higher that is a size. So, in case of drug we can use, in case of drug that is whose molecular weight is less than 500 Dalton, we can use drugs. So, that is why if you use HPC and if you do such modification like nanotechnology based therapy, in that case we can solve all those problems, but it will start with this in silico design. So, it is a task, or it is a suggestion to all the young pharmacy students, that in future if you may focus on in silico or vaccine based that is a HPC based work, you can get lots of such kinds of advancement. So, thank you all of you, thank you for joining this lecture. We may meet in our next lecture. God bless.